When holiday season rolls around, cards and messages arrive with family photographs from friends and relatives. Parents arrayed with the kids, maybe a pet or two. And in those pictures is a popular topic of conversation and curiosity. How is it that children of the same parents can end up so different from each other, and sometimes even from their parents? The answer to this question involves a process called genetic recombination, which has the effect of assuring that no two individuals will have the same DNA unless they are twins derived from a single fertilized egg. In this lecture, you'll learn how recombination has increased the number and variety of genes we have, how recombination repairs breaks in DNA, and how some impressive technologies have arisen from our understanding of recombination and repair of DNA breaks. One of these technologies for editing genes with unprecedented ease and precision has been called the most important invention since the Internet. It has already proven to be a game changer for medicine, agriculture, and research. To grasp how it works, we must understand recombination. Remember that each child and each parent, in short, each individual, has two sets of chromosomes, one inherited from each of their parents. So mom cells have 23 chromosomes from her mother and 23 from her father. Likewise for dad. In both sexes, the cells that make the eggs or sperm undergo a special kind of cell division called meiosis. This reduces two sets of 23 chromosomes to one set of 23, resulting in what is called a gamete. Fertilization, of course, will unite the egg and the sperm gametes, and that returns the chromosome number to 46. In the regular cell division of mitosis, the chromosomes are duplicated, then partitioned to two cells so that each daughter cell gets a full set of 46. Gamete formation uses a different strategy for cell division called meiosis. In meiosis, in our cells, chromosomes are doubled initially, as in mitosis, to get 46 pairs of chromosomes, but after this, the cell goes through two cell divisions without any more chromosome replication. Now, one cell division would leave us with two cells, each with a full set of 46 chromosomes, as in mitosis. But a second division without further chromosome doubling results in four cells, each with a single set of 23 chromosomes. This is how eggs and sperm come to have half as many chromosomes as the rest of our cells. There are two features of meiosis that ensure that no two kids who aren't identical twins will have the same DNA. First, remember that mom has 23 chromosomes each from grandpa and grandma, so she has two each of chromosome 1, two of chromosome 2, and so on. When mom cells undergo meiosis to make eggs, the eggs randomly receive one of the two chromosomes from each pair. So an egg may receive grandpa's chromosome 1, but grandma's chromosome 2, and so on for all 23 of the chromosomes. Theoretically, it's possible for an egg to receive all 23 of mom's chromosomes that came from grandpa, or all 23 from grandma, or any combination in between. A quick calculation reveals the number of possible permutations for two sets of 23 chromosomes. It's 2 times 2 times 2 for 23 times. And that turns out to be over 8 million. Yep, 8 million different combinations of the chromosomes just from mom. So although eggs will have one complete set of 23 chromosomes, the likelihood of any two eggs having the exact same combination of chromosomes is vanishingly small. And the same math applies to dad cells. So one egg and one sperm are each drawn at random from their own 8 million possibilities. Now together there are 8 million possibilities times 8 million possibilities, and that's 64 quadrillion possible offspring possibilities. That mind-boggling figure would be overwhelmingly more than enough to make everyone's genetic inheritance unique, even if every single human being from all of human history had had the same two parents. But there's even more. We've done the math assuming that the chromosomes you inherited from your mother and father were the exact same chromosomes they inherited from their parents. That, it turns out, is not true. A second factor increases gene shuffling even more. During meiosis to create an egg cell, mom's chromosomes swap regions or recombine the genes she got from her mom and dad. This same kind of swaps happen when sperm are created. These swaps within the maternal and paternal lines multiply the possibilities even more. To be clear, all the swaps and recombination take place within some very clear rules about what's permitted and what's not. The genes on each chromosome are all organized the same way, and that organization doesn't change. Chromosome 1 from grandpa carries the same genes in the same places as chromosome 1 from grandma. Only the versions of those genes can be different. For example, whether someone has freckles or not is controlled by the MC1R gene, which is always at the same location on the long arm of chromosome 16. Likewise, whether you have sticky or dry earwax is determined by the ABCC11 gene, which is found at another spot on chromosome 16. During meiosis, before the first round of cell division, there are 4 of every chromosome lined up, so that all 4 copies lie alongside each other. This structure, called a tetrad, allows the chromosomes to swap regions without violating the rules about where the genes are located. When they're swapping during recombination, the organization of the genes remains the same. It's like if you had 2 rulers lined up next to each other, one red and one green. If you swapped out the section between the 3-inch mark and the 6-inch mark in the 2 rulers, you'd have a red section in the green ruler and vice versa, but the rulers remain unchanged otherwise. This kind of reciprocal exchange is called homologous recombination. Since the chromosomes have different versions of the same genes, this sort of swapping shuffles the versions of certain genes present on each chromosome. So the chromosomes that parents pass on to their kids are not the exact same chromosomes they themselves carry. Each parent passes on a mashup of the chromosomes of the 2 grandparents. Recombination can happen in every chromosome, so the egg and sperm have even more diversity than is generated simply by random assignment of the original chromosomes from mom and dad. So layered on top of the 8 million times 8 million possibilities are now even more combinations. Is it any wonder that no 2 humans except identical twins have identical DNA? But how can chromosomes just swap sections? How does a DNA molecule exchange a part of its sequence with another DNA molecule? Well, let's take a look. The first thing that has to happen is a double-strand break in one of the 2 chromosomes. In meiosis, this break is created by enzymes that cut one DNA through both of its strands. Next, a nuclease acts on both the free 5' ends, chewing them back, so that both broken ends of the DNA now have overhanging 3' ends. Each of these 3' ends then invades a neighboring DNA molecule that is intact, finding matching sequences to pair with and displacing the original partner of the intact DNA. This creates a bubble in the intact strand called the displacement loop, or D-loop. The 3' end of the invading strand gets extended by a DNA polymerase, and the polymerase copies the intact DNA strand. As DNA synthesis proceeds, the D-loop becomes larger, till the displaced strand bumps into the 3' end of the broken strand. At this point, the displaced strand of the intact DNA is able to base pair with the 3' end of the broken strand. Now the displaced strand can serve as a template to extend the 3' end it is paired with. The newly synthesized regions are then joined to the broken ends, forming a structure called a Holliday junction, named for Robin Holliday, the scientist who discovered it. What you are seeing at this stage is the 2 DNA molecules are connected by crossing strands at 2 places. To separate the 2 DNAs, the crossed strands have to be cut and rejoined. This can happen in one of 2 ways. The strands that are cut could be ones involved in the original exchange or the other pair. So depending upon which way the cut is made, the the remaining DNA pieces will be rejoined to give slightly scrambled combinations of the two original strands. So information in two adjacent DNA strands can be swapped during meiosis. And two chromosomes can undergo recombination at multiple spots, so there's even greater diversity generated. Many of the same proteins responsible for recombination are used for another crucial cellular function: repair of double-strand breaks in DNA. Double-strand breaks are among the most dangerous forms of damage that happens to DNA. When a DNA molecule is broken on both strands, the two pieces of double-strand DNA can separate. This double-strand break fragments the DNA and could lead to cell death. So it's really important to ensure that breaks are promptly fixed. These double-strand breaks, or DSBs, can arise from chemical damage or cellular malfunction, but they must be fixed or they can be lethal. In a single-strand break, the problem is fixed by copying the undamaged strand. But when both strands are broken, this is not an option. What can cells do? One solution is to use a quick and dirty repair mechanism called Non-Homologous End Joining, or NHEJ. It's non-homologous because the ends of the DNA don't have to match. The idea is to grab the ends of the broken DNA and reconnect them using a ligase enzyme. But this kind of end joining often chews back the ends of the broken strands before rejoining them. As a result, when the broken ends are ligated back together, there may be deletions or insertions of bases. So non-homologous repair is fundamentally different from recombination. It does not return the DNA to its original state and is not an ideal way to make a repair. But that's where recombination can help. Just like in homologous recombination during meiosis, chromosomes are deliberately broken by nucleases that introduce double-strand breaks. And how did we end up with perfectly good chromosomes again? Through using a matching chromosome lined up perfectly to provide a template to copy. Why not use this same mechanism to fix broken chromosomes? Well, cells do indeed repair double-strand breaks by a mechanism called homology-directed repair. This is very similar to meiotic recombination, and this allows double-stranded breaks to be fixed perfectly. But chromosomes are not always conveniently aligned, so at other times cells have to make do with non-homologous repair. So we've seen that recombination during meiosis creates enormous diversity in the gametes. No two individuals have exactly the same DNA, even if they have the same parents. Recombination also plays another vital role in our bodies. It helps to generate the enormous variety of antibodies we need to fight off invaders like bacteria and viruses. Antibodies are defense proteins made in vertebrates that bind tightly and very specifically to targets like microorganisms or foreign molecules. Binding of antibodies to the intruders directly inactivates them or marks them for destruction. Our bodies make billions of different antibodies, each specific for a particular target or antigen. How can we make billions of proteins that are different with fewer than 25,000 genes in the entire genome? Well, let's think about how you could put together many different outfits from just a dozen different shirts, 10 pairs of pants, and 4 pairs of shoes. Instead of buying hundreds of outfits, you'd have to mix and match the pieces to make many distinctive combinations. And that's what immune cells do. There isn't a separate gene for each individual antibody. Instead, the antibody genes are broken up into chunks or segments, and there are many different versions of each segment. Using a special kind of recombination unique to somatic cells, immune cells create different combinations. Each cell picks one segment from each category and joins them together to make its own variant antibody that recognizes and binds to one very specific target. Given the number of choices of each segment, immune cells could potentially make about 300 billion different antibodies. Somatic recombination depends on the activity of a number of enzymes that help with the double-strand breaks and rejoining necessary for creating the hybrid antibody genes. A lack of any of these enzymes can lead to severe combined immune deficiency, or SCID, also known as bubble boy disease. Children with this condition are unable to fight off infections and may die as infants if they are not treated promptly. The best chance for such kids so far has been a bone marrow transplant from a matched sibling before the age of 3 months. So we've seen that recombination generates diversity in gametes as well as in our immune cells. More surprising is that recombination also plays a crucial role in fixing double-strand breaks in DNA accurately. An important point to keep in mind for both recombination and repair is that similarity of sequences is what allows the initial base pairing of the broken strand with one of the strands of the intact DNA. Homologous recombination then is based on sequence similarity. On the other hand, the cell's crude repair of double-strand breaks in DNA using non-homologous end joining suggests a way to deliberately inactivate genes. After all, the process causes deletions and insertions, and that can be enough to inactivate the gene. For researchers, knocking out a gene makes it possible to find out what role it plays in the organism. The challenge, of course, is finding a way to create double-strand breaks in the gene of interest at just the desired spot. This was the sticking point until the advent of a method that made it so easy to inactivate or edit genes that an entire genome's worth of mutants could be made in short order. And the name of this game-changing method is clustered, regularly interspaced, short palindromic repeats. But everybody abbreviates that to CRISPR, pronounced CRISPR, like the veggie compartment in your fridge. And CRISPR and its cousins are likely going to remake the world. But first, some background. This new tool to inactivate or edit genes started with a sort of immune system for bacteria. What do bacteria need an immune system for? Well, it turns out that bacteria can be attacked by viruses called bacteriophages, or phages for short. To protect themselves against phages, bacteria have a clever system to recognize a virus that has attacked them before. They store pieces of the virus sequence so that they can ID the virus if they see it again. The pieces of virus sequence are stored in the bacterium's own genome, separated by short repeated sequences of bacterial DNA, kind of like placing the fingerprints of each criminal in a separate file folder. The short repeated sequences are palindromes. In English, a palindrome is simply a word that reads the same backwards and forwards, like radar. In DNA, what palindrome means is that a sequence on the one strand is the same as the sequence on the complementary strand, read in the opposite direction. For example, this strand on the top reads GGA-TCC. The bottom strand reads, if you start at the other end backwards, GGA-TCC. Thanks to the good filing system made possible with short palindromic repeats, the bacteria are ready during a subsequent viral attack. The sequence of the invading virus can be compared with the collection of stored sequences, and an enzyme is dispatched to destroy any sequences that match the stored fingerprints. How? If the bacteria have stored sequences that came from a virus, the bacteria can copy the virus sequence into an RNA called a guide RNA. Because the sequence of the guide RNA matches part of the sequence of the attacking virus, the guide RNA can base pair with the viral sequence. And that's what it does. It hones in on the matching sequence on the virus. But attached to the guide RNA is an enzyme called Cas that can then cleave the virus genome. So the clustered, regularly interspaced, short palindromic repeats, or CRISPR, are the fingerprint files. Cas is the CRISPR-associated enzyme that cuts up anything that the guide RNA base pairs with. Cas9 refers to the Cas enzyme discovered to have the desired effect. It came from a particular bacterium, Streptococcus pyogenes, which was chosen for closer study because of its relatively simpler CRISPR system. This is why you often see CRISPR referred to as CRISPR-Cas9. Okay, that's good for bacteria, But what's all the fuss about a bacterial immune system? The big excitement is that we can use this same system, but paired with any guide RNA we can design. Since the guide RNA is what determines which sequence gets cut by Cas9, all we have to do is design guide RNAs that base pair with the gene sequence we want to target. Basically, we steal the Cas9 enzyme and pair it with a guide RNA that will take it straight to the gene we want taken out. It works in any kind of cell, not just bacteria. This RNA-guided Cas9 missile is fast, easy to deploy, and amazingly effective. What it does is make a double-strand break in the DNA that the guide RNA leads it to. It can make double-strand breaks in any DNA, not just viruses, as I said. And you know what happens when there's a double-strand break. The cell rushes to fix it. If it's timed right, when the cell isn't preparing to divide, the double-strand break will be fixed by non-homologous end joining, which, as you saw earlier, results in deletions and insertions. Such sloppy repair has the benefit of rendering the target gene inactive. So all it takes to inactivate a gene is to design a guide RNA that will base pair with it and couple it with Cas9. Deliver the system into a cell, sit back, and let your guided missile do its thing. Researchers could now create knockouts of every gene in a genome one by one, or in combination, to find out what role each one plays. First-generation CRISPR works well to knock out genes, but sometimes there are off-target effects. For example, if a guide RNA mistakenly binds to a sequence that is similar but not identical to the intended target. Scientists are working on improving the specificity of the targeting by using Cas enzymes that are more accurate than the original Cas9. Researchers are also trying to perfect a CRISPR-like system that would deliver an enzyme that can edit a single base in DNA. Such a system would bring a base editor to the site of a mutation where it would fix the error rather than making a double-strand break that would need to be repaired. One such editor enzyme has already been tested in the laboratory, but is not quite yet ready for prime time. If this method is optimized, it would enable researchers to easily correct point mutations like the one causing sickle cell anemia. So missile strike knockouts that destroy a target are just the beginning. CRISPR and related techniques can do a lot more. For one thing, it's possible to fix a double-strand break made by CRISPR using homologous recombination too. But instead of having the break be fixed by using another chromosome, as in meiosis, it is possible to provide a modified form of the desired gene as a template. So the break is corrected by using the provided DNA, which would be a particular variant of a gene. If this route is used, then the version of the gene originally in the chromosome is replaced by the version that is provided. Imagine what this would mean for genetic disease. A faulty gene could be replaced with a good copy. A variation of CRISPR depends on completely inactivating the Cas9 enzyme. Now, this technique is not aimed to edit genes. Instead, it relies on using a guide RNA to send the Cas9 protein to, say, a spot in front of a gene's promoter, where it would bind and prevent transcription. This could be used, for example, to block the expression of normal genes associated with cancer or with other diseases. There are so many new applications for CRISPR coming out that it is impossible for us to go into all of them. But one called a gene drive merits attention because it changes the way genes are inherited. Normally, as you know, the chance of a particular trait being inherited is 50-50. What a gene drive does is to essentially increase that to 100%. This can force a particular version of a gene to spread rapidly through an entire population. One reason gene drives are being studied is to wipe out the mosquitoes that carry diseases like malaria, Zika, or dengue. In the past, genetically engineered mosquitoes have been made with the idea of spreading a gene that interfered with the reproduction through the population. But eventually, over time, the gene would get diluted out as the engineered mosquitoes bred with normal ones. With a gene drive, there would be no dilution, and the gene would spread to the entire population in only a few generations. But how does this miraculous gene drive work? Researchers genetically engineered a male mosquito to have a mutation in a gene called double sex that plays a role in sex determination in mosquitoes. Researchers also inserted the genes that encode Cas9 and the necessary guide RNAs to knock out the normal form of the double sex gene. Females with one copy of the double sex mutation can reproduce normally, so they can pass on the genes. But if they have two copies, they behave like males and lay no eggs. When an engineered mosquito is mated with a normal mosquito, the offspring will have one normal version and one mutant version of the gene. In this mosquito's cells, the guide RNA will direct the Cas9 to cut the normal version of the gene. And you know what happens next. The mosquito's cells will try to fix the normal version. In cells undergoing meiosis, this repair happens by copying the other chromosome, which has, drum roll, the mutant version. So now both copies are the mutant version, which means all the gametes will carry the mutation. When this mosquito then mates with one carrying the normal version of double sex, its progeny will have the same thing happen. When enough females have two copies of the mutation, the population will crash. In 2018, scientists at Imperial College London showed that a population of mosquitoes could be wiped out in 8 generations using a gene drive. Researchers are cautiously optimistic that it could be used in areas where malaria is endemic. Thanks to the guide RNA, any gene drive is very specific to a particular sequence of DNA. And before carrying out any targeted releases in the wild, these same researchers are working to devise gene drives that can reverse the original one should that be needed. If equally successful in the field, this approach could save 500,000 lives each year. So our understanding of genetic recombination and repair are leading to technologies whose power seems unlimited. Scientists have already used CRISPR in laboratory settings to correct genetic defects. Hearing has been restored to deaf mice, and the mouse versions of type 1 diabetes and a liver disease called hereditary tyrosinemia have been successfully reversed. Dogs have been cured of Duchenne muscular dystrophy. In non-viable early human embryos, attempts at curing beta thalassemia, hypertrophic cardiomyopathy, and Marfan syndrome have had some success. Beyond medicine, CRISPR has been used to make wheat and rice resistant to fungal pests. Oilseed yields have been doubled. Mushrooms have been modified to last longer without browning, and pigs have been engineered to be leaner, for those who'd like CRISPR bacon, perhaps. Projects are underway toward superior varieties of corn, tomatoes, sugarcane, and strawberries, and lawns that'll require less mowing. Yeasts and bacteria have been engineered to produce biofuels and bioplastics. The sheer power of these techniques calls for a discussion of how to use them safely. In 2018, a Chinese scientist, apparently acting independently of his institution, announced that he carried out gene editing on human embryos. In March 2019, some of the leading researchers in the field called for a global moratorium on any kind of human gene editing that can be inherited to provide time for an open discussion on the ethical and safety considerations of such procedures. Ready or not, we're going to have some conversations about the use of these technologies that will be reshaping our world.